CUG or CUA. Basically, instructions for making the same protein can be written with different like spellings, so different codon usages. And so we can optimize the codon sequences in order, so we can change the spelling without changing the actual protein, and we can optimize this in a way that is specific for the types of cells that we're trying to get a protein expressed in. So to get these cells to make a protein for us, we can optimize the sequence, optimize the like the DNA sequence and then the corresponding messenger RNA that gets made into the protein. We can optimize that sequence in a way that's going to make the cells hopefully express more of it and this protein be more stable and soluble and have better yield. And so we call this codon optimization. And so here's an overview, and then I'll go into more depth. Just how gray with an A and gray with an E are both mean the same color, but you're more likely to find the gray with an A in American language and gray with an E in English, in like British language. You're more likely to find CUG and CUA both like spell the same amino acid, leucine, but you're more likely to find CUG in certain organisms than CUA. And so if you were to if a publishing house was to publish a book and they wanted to publish this, they might publish like an American English version and a British English version. And they would put the gray with an A in the American version and gray with an E in the British version. Similarly, if you want to get a protein expressed in a different organism that it's normally expressed in, so say I want to take the instructions for making a human protein and stick that into bacteria and get the bacteria to make the protein for me, I'm going to want to use like the dialect or whatever that the bacteria normally use. So although the bacteria understands that both of these spell leucine, so we'll get more into what this means in a bit, but basically these both are going to instruct the protein making machinery to put in a leucine, because the genetic code is what we call degenerate, which means that multiple codons, so these 3-letter RNA words, can spell for the same amino acid. So both of these are going to tell the ribosome, so the protein making machinery, to put in a leucine. However, the different organisms are going to stock up on different levels of the tRNAs that are going to bring the corresponding amino acid. So even though the cells are capable of knowing that both of these can mean the same thing, it's like easier or more normal for them to use one of them. And so if we want to get those cells to make a protein for us, we want to use like the dialect that they normally use. So we want to avoid using rare codons, so things that they don't have many tRNAs for, things that they don't use that often, and instead use codons that they do use more often. We wouldn't want to use them like all the time because then we just deplete them. But you can kind of like harmonize to use like the same sort of levels as the cells normally use. But that's not all that matters. So if you were to make an American English version and a British English version of a book, you're not just going to change the spelling of like gray and honor and stuff and like put those U's in places that you see extra U's and stuff. Instead, you're going to want to change other things too, because there are like differences between the languages that we can understand one another, but we have subtle differences, especially in things like second meanings of words type of thing. And so one word might have a connotation in one of the languages that might be lead to some awkward conversations or something if you don't realize that something has a second meaning, like a kind of joke meaning or sometimes inappropriate, that sort of thing. So you'd want to avoid those sort of things. Similarly, when you are optimizing the gene you're putting into the cells, you want to make sure that you're not accidentally or introducing a sequence that looks like something else or that has a kind of like different meaning. So you don't want to put in a sequence that's going to look like an internal ribosomal entry site or something, or like a ribosome binding site, or a premature or like a cryptic splice site or something random like that. So you don't want to put in those sort of things. So that's another layer of things you have to look out for when you're doing codon optimization. Don't worry, like companies do this all in like this proprietary method, but these are the sort of ththings that they take into account. So it's not just the codon usage bias, that's just one aspect. It's also things like this hidden sites and those sort of things. like thin shiny Delgrano sequences, cryptic slice sites, that sort of thing. So that's the second aspect. And a third aspect is you have to consider the structure of the DNA and the structure of the RNA. So it's not, when you're changing the letters, it's not just affecting like what tRNA comes. And so instead, you're also potentially changing the structures of the DNA or the RNA. And so typically, like, don't really think about their structures that much if we're like focused on this protein, but on the protein structures, especially if you're a structural biologist. But the DNA and the RNA, they can have structures too. And so if you have like regions where they have like a really strong GC rich region or something, that could influence, like make the DNA make a weird shape or make it harder to access when you're doing transcription. So your DNA has to be transcribed, so you have to make the messenger RNA copy, and then that messenger RNA copy has to be made into protein. And the translation step. So you need to make sure that you don't have any like secondary structures messing up with either the transcription or the translation. So you want to make sure, it's kind of like making sure you don't have any tongue twisters, I guess, in your book. If you want to make it easiest to read, you want to have a bunch of tongue twisters. So if we want to make our protein easiest to make, we're not gonna want to have things that introduce like knots or stuff in the DNA or make it really hard to access the transcription start site. So we wouldn't want to have things that are going to make strong secondary structures in our RNA. And so RNA, it can form like loops and stuff with itself. It likes to be single-stranded, but it can form like hairpin loops and that sort of thing. And different the ability to do this is going to depend on the sequence. And so if you want to make your sequence so that you avoid having these hairpins, avoid having these structural elements that are going to interfere with the translation. So those are just a few of the main things that go into codon optimization. And so that's just a basic overview. And now let's go look in a little more detail, as well as kind of strategies other than codon optimization. When you're not just like codon optimizing an entire gene, but maybe you want to introduce some synonymous mutations through site-directed mutagenesis. You can introduce like restriction cut sites and that sort of thing. Or you can actually just, instead of changing your sequence, you can change the cells. So like in bacteria, either like Rosetta strains that you can put in like extra tRNAs. And so that's just like a basic overview. And yeah, so sorry, I just kind of like talked this out. And so I hope I didn't lose anyone. If I did, hopefully this next part will help. So proteins are chains made up of chains of amino acid letters, and the instructions for making them are written in the form of genes, which are DNA sequences, and then in the process of transcription, one strand of that DNA is going to be used as a template to make a messenger RNA copy. That copy is going to get edited a little, and then the ribosome, so this protein making complex, is going to move along this messenger RNA. And it's going to link together the amino acids in the order specified by the messenger RNA sequence. And so the way that it reads this is it's going to read this in 3-letter words or codons. So you have 3 RNA letters are spelling one amino acid. And the way that you go between these is with a tRNA, so a transfer RNA. On one end, it's going to have an anticodon that's complementary to the codon. And it's carrying the corresponding amino acid. So in this way, the ribosome can travel along the amino acid, adding in the amino acid specified in the correct letter to get the protein. And so the sequence that you get is going to, the protein sequence is dictated by the messenger RNA sequence, which is a copy of the DNA sequence or the gene. We can stick this messenger RNA, we can stick a DNA version of this messenger RNA, which we call complementary DNA or cDNA. We can basically stick that into cells to get them to follow those instructions and make a protein for us. We call this like recombinant expression because we're recombining the genetic instructions for making that protein with like a plasmid vector or some sort of vector. So plasmid is just like a circular piece of DNA, and then
You can stick those into expression cells, like bacteria or insect cells or mammalian cells. There's different cell types, which is a key thing about codon optimization is that you can optimize the sequence for the cell types. And so the reason why you can do this optimization, why you can change the sequence, so there are changes that you can make to the sequence that you would that would change the actual protein sequence, but there are also changes that you can make that wouldn't. This is because the genetic code is degenerate. So this genetic code, the which codon goes with which amino acid. So this you can tell from like a codon table. So a tRNA is going to, with this anticodon is going to, or with the anticodon matching this codon is going to carry this amino acid, like always. And so this is why different organisms can all read the same instructions because this code is not ambiguous. One codon can only spell one amino acid or stop. But it's also degenerate. So you can see in this codon table that like leucine, it has 6 different different codons that all spell leucine. So this genetic code is degenerate, but not ambiguous. It's also not overlapping. So it's going like 3 by 3 by 3. It's not going like overlapping, or else you would get this weird stuff. And the right one is signaled by a start codon. So you can imagine that there are changes that you can make to the sequence that would change the amino acids. We call, if you have like a point mutation that changes a single letter, we call this like, it can either be like silent or synonymous, where it basically changes to the same amino acid. So it doesn't change the amino acid you make because of that degeneracy. Or it could be missense, in which case you have like it's changing. If it's non-synonymous, it's changing the amino acid. So silence kind of I guess a less specific, more specifically, it would be a synonymous mutation, not necessarily silent because there could be effects on the mRNA structure or how it's read or that sort of thing, which is kind of getting into the points of we'll get into with the codon optimization. You can also have mutations that make insertions and deletions and stuff or in premature stop codons, but we're not going to get into that. What we really care about is these point mutations. And so we'll think about the synonymous mutations, which are going to leave the letter, the protein letter the same, and the non-synonymous mutations that are going to change the letter. And so we can imagine changing the DNA letter and then the corresponding mRNA letter without changing the protein letter. These are called synonymous mutations, and they're the basis of the codon optimization strategy, is that we're going to introduce synonymous mutations. So we're making mutations in the DNA and the corresponding mRNA, but not the protein. And we're going to optimize it for the cell type that we're going to want to express it in. And so there are different things that you need that go into optimization. One of them, and kind of the most like obvious, I guess, or simplest on this is it used to be thought that this was like the main thing, but now people are recognizing that there's actually a lot going on other than just the codon usage, and that codon usage itself, the effects on translation and things are kind of tricky, and protein expression are more complicated than it would originally seem, but still codon usage bias is something that is a factor, just not the only factor. And the idea here is basically you want to avoid rare codons. There could be less of the corresponding tRNAs and the ribosome could stall, but you don't want to change all of them because then you're going to deplete those ribosomes. So you want to use like a codon frequency that's found in the expression cell type. So let's look a little bit more about like at an example type of thing. So I had this graphic or whatever was from a couple years ago, but I was looking for like a real life example of a peptide, but because I wanted to do like show like the sequence. And so this is a really short fruit fly peptide. So it's like a short protein. It's called TAL. And it shares an mRNA with a few other peptides. It was like a polycistronic mRNA, but it gets translated by itself, so it has its own start and stop codon. So it made a good example when I was trying to show like a real sequence that would going from the DNA to the protein. But anyway, so this is a real peptide, this tau peptide from a fly. And so what's going to happen in translation is that the the tRNA is going to have to go along the messenger RNA and bring in the corresponding amino acid. And so each amino acid is going to have at least one tRNA, but as we saw from the codon usage table, some might have more than one. And so we can basically potentially optimize the sequence to give it more of the ones that it has more of those tRNAs for. So if we take into account the codon usage bias, so if we were to look at this sequence, we can see that these two alanines use two different codon spellings. So this is an example of your degeneracy. And they might use the C and tRNA because they're only different at the wobble position, the third letter in the codon. So this is a thing, there's like the third letter, there's also like this importance of this, like the what letters in the third position. It's like the wobble position, and there's some stuff with like optimization of the wobble position. But anyway, I'm not going to get into that. But some tRNAs actually some tRNAs can use multiple, go to multiple sequences, but it's always going to spell the same thing. So they might use different tRNAs. And so then these, but let's look at these. So that's an example of the degeneracy already at play, but let us look at the, but let's look at this one, the tyrosines. So there are these two tyrosines. So tyrosine has the abbreviation Y. Y, you might ask, well, T is for threonine, and the Y is the second letter in tyrosine. So there you go. But so it uses, this one uses, they both use this UAC codon. And so, but is it the optimal codon? So what are our options? So we can look at a codon table, a codon usage table. Or let's first look at a codon table. So if we look at tyrosine, we can see that there are two anticodons for tyrosine, and they differ in the third position. So they might still use the same tRNA, but we'll assume they have separate ones for this illustration. Not exactly sure. They kind of vary from organism to organism and things, and yeah. But which one is optimal for our expression system? So you can see tyrosine, our options are UAU and UAC. In the Drosophila sequence, it has this UAC, which makes sense because in Drosophila, they use UAC 63% of the time and UAU only 37% of the time. But if you want to express it in a different cell type, then those cells will have different preferences. So if you are going to express it in bacteria, say, you might get a little better results with swapping it, but not likely to make a big change. And similarly, you seem to have pretty equal levels with most of them. But with insect cells, you can see that basically things are swapped. You have 71% uses the other, and 29%, only 29% uses the one you have. So if you were to swap these, then you might get better results. So that's just an example of like a single site. Similarly, if we were to look at, say, leucine, so E. coli uses, if we look at a codon usage table, and so you can see like these, if you just Google these tables, like this one's from biologicscorp.com/tools/codon usage, but there's a bunch of different ones you can find. It's pretty easy if you just Google it. So E. coli, you can see that these are the 6 codons that spell for leucine. And this is the codon usage table for E. coli. So this is showing you the E. coli preferences. And so it's showing you like the amino acid, the codon, the amino acid, 1-letter code, the fraction, every time it's used. So you can see that E. coli, it's going to use CUG the most. So it's like 0.5, the fraction of 0.5, and CUA the least. So it has a 0.04. So if you're trying to express a protein with lots of CUAs and it's mRNA, like the ribosome might stall if it has trouble finding a matching leucine tRNA because the cells aren't like stocked up with that tRNA. And so basically you can use codon optimization for the codon usage bias to change the spelling of the gene to match the one that the cells like. And so this is this is an example of codon optimization. Or you can make the cells stock up on the ACUA matcher, so give them lots of copies of the gene for that tRNA, so get them to like overexpress the corresponding tRNA. And this is a strategy used by like Rosetta strains of E. coli. And so they have like an extra plasmid that's going to get them to make more of this tRNA. And so that it will incorporate the leucine from that codon more efficiently without stalling. And so when the tRNA is coming along, it's going to be easier, or when the ribosome's coming along, it's going to be easier for the corresponding tRNA to be found. So this is an example of the like codon bias optimization, but that's not the only thing that gets optimized for. And so, for example, if you were to go like GeneScript or GenScript is like a main one, so like GeneArt and stuff that do codon optimization, but they optimize for a lot more things than just the codon usage bias. And so some other things that are optimized for are the you need to consider the codon in its context. And so when we're looking at this, when we're looking at like a single site, we're basically just focused on that single site. And maybe if you want to make a single mutation, like sometimes if we're doing site-directed mutagenesis, so we're changing like a single letter and actually want to change a letter, like to a different letter, like we want to change the protein letter, we want to make an amino acid substitution. In that case, we need to like go to a codon for a different amino acid. But we have basically, if we were to want to introduce a leucine where there was normally like a valine, or there was normally some, I don't know, something else, what we would do is we could go to a table and see, okay, well, which one would be best to put in? Like you wouldn't want to put in the CUA for these, if you were doing it in bacteria, like the CUA, you would instead want to put the CUG, because now you get to basically choose from all of these. And so that's sometimes I look at a codon usage table when I'm making site-directed mutations. In that case, I'm just basically thinking about like a single amino acid. But it's also important to note that like when we're doing this in whole genes, not only we'll talk more about the context, but also think about like this is not the only place that you'll be changing it. So you wouldn't want to change all of them to CUA, or else then you're going to run out of that one, right? Or if you change all of them to CUG, because then it might run out of that one. And so then there's this idea of like harmonizing. So you basically use the frequency of, overall you use the frequency of the cell type. There are also other things though. So we need to think about the codon in its surrounding context. Thankfully, we don't like, people aren't just typically like thinking like designing this themselves. Like there's those software tools that use all these algorithms. These are just some of the things that these algorithms are like taking into account. And so the codon has to be considered in its context. And so basically it's not alone. And you have to make sure that like when you're making a change, it's not introducing a sequence that resembles some sort of like functional motif, something like a ribosome binding site or something like inside of the protein that's going to make you have like get like a truncated protein made, or that sort of thing. Cryptic slice sites, that's shine diagonal sequences, Tata boxes, various things like various sequences that the change you make might make the sequence resemble something that the cell is like, oh, hey, I know what that is. I should do something here. And you're like, no, I didn't mean to do that. Like, we don't want to do something here. There's also, you want to watch out for secondary structure. So you have to consider that you're not just affecting, so basically we're interested in this protein, but it has to go through this, like it's going to be in this gene form, this double-stranded DNA, and then it's going to be in this mRNA form, and both of these can adopt like secondary structures, especially the mRNA, that can influence how accessible the site is, say for ribosome binding or for the translation to happen efficiently. So you want to watch out for those secondary structures, things like hairpins and loops and that sort of thing. And so this software takes that into account as well. And I am not advertising for GenScript. I don't get paid for GenScript. This is just one of the common ones that use, and this is their website of the type of things that go into their thing. Also see with like optimization things about like uridine depletion. And so basically uridines, if the RNA has a bunch of uridines, it can trigger like an innate immune response in like certain types of cells and stuff. So this isn't like a you don't have to worry about this, like if you're expressing just like a bacteria or whatever. But when people are doing things like mRNA for vaccines or protein expression in humans, then having a bunch of uridines can be a problem because it's setting off like these, it's binding to these like receptors and setting off these innate immune responses. And so actually figuring this is like, well, specifically unmodified uridines. So in our cells, a lot of uridines actually get modifications, like pseudouridine modifications or other modifications. And so one of the key findings of Catalin Carrico, who was like one of the key people behind like mRNA vaccines, was actually finding that modifying the uridines actually decrease the immunogenicity, so the cells wouldn't just like degrade the mRNA that you put in. And so that's what the whole like uridine depletion thing is that they can make it so that if instead of modifying the uridines, which they can use like synthetic uridines or whatever, but if you want the cells to make more of it and that sort of thing, you can't really just do that if you're not putting in the mRNA, if you're putting in the gene. But you can change the sequence to avoid having a bunch of U's. And so that's the whole uridine depletion thing, if you get confused about that. And a cool connection to things today. They also had a good webinar that I'll link you to, which also pointed me to this cool paper. This was published in 2009 in Science, Kudla et al., coding sequence determinants of gene expression in E. coli. And basically they did, they made a synthetic library of genes for GFP. So these had a synonymous, random synonymous mutations. So they were changing the DNA and the mRNA without changing the protein. And they then expressed them in E. coli, and because you have GFP, it's fluorescent, so it will shine and you get a readout of how well it's made. And they found that codon bias did not correlate with gene expression, but instead it corresponded that the codon bias influenced the global translation efficiency and cellular fitness, so it made the cells happier, but it didn't increase the expression of that specific gene specifically. And so instead, they found that the biggest determinant was actually the stability of the mRNA folding near the ribosome binding site, which can explain more than half the variation in protein levels. And so there's a lot of things that go into the expression of a protein. Another thing that they were talking about in that webinar I heard is that like the sequence of the, like sometimes your cells like purposely use a rare codon, and it can help with like protein folding and that sort of thing. If your protein, like if it needs to kind of stall a little bit at a certain point in order for the part that's come out of the ribosome to kind of like fold up and that sort of thing. So the codon usage can also influence things like functionally important things, which is one of the reasons why instead of like calling things like silent mutations, we should instead say synonymous mutations. Although silent is commonly still used, and that's why it was on that slide, but I'll fix that. And so synonymous mutations, because they're not changing the protein, but they can be changing other things. And this is also just like a really fascinating thing, is that a lot of the focus is on changes that non-synonymous mutations, so things that change the protein. And that's kind of like easier to conceptualize and to think about and to test the importance of various things. But it's also important to note that like synonymous mutations can also have consequences on things like protein expression, things like splicing, and various processes in cells. And so, all the stuff about like junk DNA and that sort of thing. So you could also have changes in non-coding regions. So you don't even talk about having a synonymous mutation there because there's no protein being made. But these sequences can still make binding sites and various things. So there's a lot of cool stuff going on in mutations, not like cool stuff, but there's like, that sounded really weird. But there's a lot of interesting biology that happens when you have changes in DNA in sequences that aren't actually making proteins. And so this is like a lot of like evolutionary stuff and really cool regulatory stuff, various cool things. And so not just bad things. And so, yeah, just like something to be aware of. Another thing to be aware of is, so we can also sometimes introduce synonymous mutations in order to introduce or remove, if you don't want one there, a restriction enzyme cut site. And so restriction enzymes are these site-specific DNA nucleases that are going to basically recognize a specific DNA sequence and bind and cut. And so we use these a lot when we're doing like cloning sometimes, or when we want to test things, like whether the thing has a site or not. And so sometimes when we're doing cloning or whatever, we want to introduce a site so that we can get it to cut, so we can then like stitch pieces of DNA together. And so we can introduce or remove site recognition sites by changing the DNA letters, but without changing the protein. And so this is another strategy. And there's this like tool called like WhatsApp, or not WhatsApp, WhatCut, WATCUT, which helps you find like cut sites and mutations that you can make, synonymous mutations you can make, in order to introduce cut sites for various restriction enzymes. Another last thing is, so basically this is a codon optimization is one tool for increasing expression, but it's not guaranteed, and it can be expensive. And so there are a lot of other things you could do as well to try to improve protein expression or solubility, and I have more posts on that. But codon optimization is a key way, and sometimes you do so much troubleshooting that you just go, like, you're going to be doing a bunch of troubleshooting or that sort of thing if you don't optimize it, and then you eventually think you're going to end up optimizing it. So like I just ordered an optimized version of a gene, and so for like insect cell expression, because it's not like normally expressed in insect cells, and so I want to give it the best chance of success. And thankfully my lab has the resources to be able to do that, and it would probably end up being cheaper than trying to express it over and over in ways that don't work with different mutations and stuff. But there are different things that you could do. If your protein, one of these is like adding a fusion partner, which can help kind of like trick the cellular machinery into making the protein. So you basically, in front of your protein, you put a protein that the cell likes making, and you kind of like trick it into making it, and that also helps with the solubility. And so there are various things that you could do to increase the expression and your chances of success. If you can't afford codon optimization, or even just like if codon optimization doesn't work, or if you want to try, it's like, I mean, like even with codon optimization, you're probably going to do a bunch of other optimization often in order to get protein expression to work, but it's an important thing.
Video này do mình, Cương bên npslab.vn, tự chỉnh sửa phụ đề hoặc lồng tiếng bằng công cụ, thấy vậy thôi, chứ tốn công lắm á. Thấy hay thì nhớ bấm theo dõi kênh, cho mình thêm động lực làm mấy video phòng lab xịn sò nữa nha.